My dear students of 11th class, and today in your botany we are going to discuss osmosis. And previously, in our previous classes, we have taken up the transport in plants, and in our previous class we have discussed the water potential, and today we will take up the osmosis. You must have heard the term osmosis. It's a special type of diffusion, and now please stay keen to understand what osmosis is. The plant cell is surrounded by a cell membrane and a cell wall. The cell wall is freely permeable to water. That means it allows the water easily to pass through. Cell wall allows the water to easily pass through. It is permeable to water and also to the substances in solution. The substances which are dissolved in the water also get through the cell wall easily. Hence, is not a barrier to movement. This means that the cell wall is not providing any oppose to the movement of either the solution or the water through it. And in plants, the cells usually contain a large central vacuole whose contents, the vacuolar sap, contribute to the solute potential of the cell. You must be remembering the solute potential. And in plant cells, the cell membrane that lies beneath the cell wall and the membrane of the vacuole, the tonoplast, together are important determinants of the movement of the molecules in or out of the cell. Now, osmosis is the term used to refer specifically to the diffusion of water across a differentially or semi-permeable membrane. Again, osmosis is the term used to refer specifically to the diffusion of water across a differentially or semi-permeable membrane. Osmosis occurs spontaneously in response to a driving force. The net direction and the rate of the osmosis depends on both the pressure gradient and the concentration gradient, which we have discussed previously. Water will move from its region of higher chemical potential or concentration to its region of lower chemical potential until equilibrium is reached. Again, water will move from its region of higher chemical potential or concentration to its region of lower chemical potential until equilibrium is reached. At equilibrium, the two chambers should have the same water potential. And in your earlier lower classes, you may have made a potato osmometer. At some earlier stage in the school, if the tuber is placed in the water, the cavity in the potato tuber containing a concentrated solution of the sugar collects water. It is because of the osmosis. And now, let us discuss another experiment where a solution of sucrose in water taken in a funnel is separated from pure water in a beaker through a semi-permeable membrane. Here we use a thistle funnel that is covered with a semi-permeable membrane, and again we can use an apparatus like this so as to maintain the pressure within the thistle funnel that is covered with a semi-permeable membrane. And now you can get this kind of membrane in an egg. The egg membrane is a semi-permeable membrane that is easily available. We can remove the yolk and the albumin through a small hole at one end of the egg, and then place the shell in dilute solution of the hydrochloric acid For a few hours, the eggshell dissolves, leaving the membrane intact. And now, water will move into the funnel because of the higher water potential outside the funnel, resulting in a rise in the level of the solution in the funnel. And this will continue till the equilibrium is reached, so that the internal and the external environment, that is the concentration, is equal within the thistle funnel and in the beaker in the surroundings. In case sucrose does diffuse out through the membrane, will this equilibrium be ever reached? No, never. External pressure can be applied from the upper part of the funnel such that no water diffuses into the funnel through the membrane, and this is to make you understand what the osmotic pressure means. So, external pressure can be applied from the upper part of the funnel such that no water diffuses into the funnel through the membrane, and that is here on the right hand side B diagram. The pressure can be maintained externally so that no water enters inside the funnel. This pressure required to prevent water from diffusing in is in fact the osmotic pressure. This is the osmotic pressure, and this is the function of the solute concentration. More the solute concentration, greater will be the pressure required to prevent water from diffusing in. Hope you may understand this. Again, I will repeat this. Greater will be the pressure required to prevent water from diffusing in. And numerically, osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. Osmotic pressure is the positive pressure applied, while osmotic potential is negative. And this is the experiment here, the demonstration of the osmosis. The thistle funnel is filled with sucrose solution and kept inverted in a beaker containing water, and water will diffuse across the membrane as shown by the arrows in the diagram A. To raise the level of the solution in the funnel, and pressure can be applied as shown to stop the water movement into the funnel as represented in the figure B. Hope you may have understood this, and this is very important in terms of your examination. Thanks. Have a nice day.